We are at a critical point in Kentucky when it comes to vaccine policy. Vaccines prevent illnesses and deaths of Kentuckians, so much so that the success of vaccines has virtually eliminated diseases such as polio, measles, and mumps. Generations of citizens have never witnessed the devastation and lifelong effects of these and other deadly infections. We are blessed as a nation to have life-saving vaccines so readily available to our citizens. We cannot and we should not take for granted the scientific breakthroughs that allow such innovation, protecting the health of infants and children and into adulthood. Life expectancy in the United States has increased dramatically with the availability of vaccines. Many nations and people across the world are not so fortunate, and we see the concerning reemergence of diseases like measles, which killed more than 40,000 people and as recently as the 1960s. More than 50 years of safe and effective vaccines and the eradication of these diseases is now at risk with vaccine hesitancy and the current misinformation campaign. I urge you to talk to your doctor or your trusted healthcare provider to help you make the best decision for you and in protecting your health and the health of your family. Your decision to be immunized directly impacts the cost in healthcare dollars, taxpayer dollars, and lives. If you are a healthcare worker, talk with your patients, your neighbors, your family, and your friends about vaccines, especially the COVID-19 vaccine. Take the time to ask and address questions and concerns non-judgmentally and discuss the safety and effectiveness and the science behind vaccines. Information is powerful. It is the American ideal to protect each other, to look out for the best interest of your family and your neighbors. If you don't have your health, nothing else matters.
Good afternoon, and thank you for joining us again this afternoon for the continuation of the Howard L. Bost Memorial Health Policy Forum. Next, we're going to hear from Erome Daniel Hankore. Dr. Hankore has a Ph.D. in chemistry and is currently a postdoctoral scholar in the College of Pharmacy at the University of Kentucky, where she studies biosynthesis of natural products that passes strong antibacterial and anti-tumor properties. Her research background includes experience in biochemistry, molecular biology, biocatalysis, or I think I have said that correctly. Thank you. Protein engineering, unnatural amino acid incorporation, and natural product research. He's also a big fan of astronomy. Please welcome Dr. Erome Daniel Hankore.
Well, thank you so much for that wonderful introduction. I hope you can all hear me well. I just want to thank you so much, first of all, for this opportunity to talk about vaccine development, research, and approval. Thank you, Bonnie, for that wonderful introduction. And it has been wonderful so far. I've learned a lot from Dr. Stack's talk, as well as the panel discussion that we've had. And my goal today is to talk about the vaccine research development and approval process in the US. And I hope that I can convince you that the COVID-19 vaccines that are currently available are very safe and very effective at protecting us from this horrible disease that we are currently trying to tackle. So here's an overall outline. I'll talk about what vaccines are, give you a little bit of an origin story, talk about how novel vaccine candidates are developed, see how they are tested in animal models as well as human volunteers before they are finally approved by the FDA for administration to the wider population. So I want to start with the history, the story of vaccines, which is intricately woven into the history and the story of smallpox disease, which has ravaged and plagued human populations for thousands and thousands of years. In the early 1900s, in Africa and in Asia, people began to practice variolation because they noticed that folks who had caught the smallpox disease did not catch it a second time. And so what they would do, they would take material from lesions of smallpox in individuals who had the disease, and then they would administer that material to healthy individuals, either through the skin or through the nose. And they found that this was very effective at protecting populations against smallpox. Now, in 1721, there was a woman by the name of Lady Montagu who was in the Ottoman Empire. She was there because her husband was an ambassador ambassador there, and she came across the practice of variolation. She herself was a survivor of smallpox. In fact, her face was disfigured from it. She had a brother who had passed away from the disease. And so she decided to get her small children vaccinated and began to introduce this practice into English society as well as the British court. But it wasn't really until the work of Edward Jenner in the late 1700s that this wonderful medical intervention that we call vaccine or vaccination was developed. So Edward Jenner learned that milkmaids who would get cowpox were actually also not susceptible to getting smallpox. So what he did was he took some cowpox material from lesions from a milkmaid by the name of Sarah Nelms, and he took that material and gave it to an 8-year-old boy called James Phipps. And then a few days later, he then exposed the little boy to smallpox and found that this little boy actually did not get smallpox. So that was revolutionary. He did the same thing with many different kinds of individuals, found out that cowpox, which was a milder and much less contagious version of smallpox, was actually able to protect individuals against this terrible disease. Now, vaccines worked, but we didn't really understand how they worked. And this is where the work of Robert Koch and Louis Pasteur becomes really important, because they helped us to understand the germ theory of disease, that there were these microorganisms that would cause infectious diseases, and that vaccines were somehow interrupting the process of severe diseases coming up once pathogen exposure had occurred. And finally, Edward Jenner's work also paved the way for our understanding of immunology, because in the late 1800s, the works of Paul Ehrlich and von Behring helped us to understand that there were these neutralizing molecules known then as antitoxins. We know them now as antibodies that were released when individuals were exposed to pathogens or vaccines like the cowpox, and they found out that this immunology, there was an immune system there that was responding to pathogens and protecting individuals. Now, in the case of smallpox, the pathogens are variola minor and variola major, and you can see the picture of those microorganisms up here in the black and white, and at the bottom you can see the disease itself. And these scabs, even in recovered individuals, remain as scars. Now, then, much as in now, unfortunately, there was vaccine misinformation. And so we had individuals who really believed that taking the cowpox as a vaccine would actually change human beings into cows. But despite all of that misinformation, there were huge campaigns, worldwide campaigns in the last few years, centuries, decades, where there was a massive campaign to get people vaccinated against smallpox. And so this was so effective that in 1980, the World Health Organization declared that our world was free of smallpox. Now, mind you, this is a disease that used to kill about 400,000 people annually. In 18th century England, the infant mortality rate was really high, 80%, even up to 90% in some areas of Europe. And yet vaccines were so effective that they essentially eradicated the disease from our planet. So what is a vaccine? A vaccine is a biological product, and it is used to induce an immune response safely in individuals. And this immune response essentially primes the individual to not get a disease. It confers protection such that there will be significantly reduced risk for severity of disease upon exposure to disease-causing pathogens. And the essential component of vaccines is what we call an antigen. And an antigen is either parts of a pathogen or a whole pathogen weakened, a weakened form of the pathogen that is used to deliberately expose an individual to that disease-causing pathogen or some part of it in order to elicit an immune response so that the body is ready to ward off the disease upon exposure. Now, you all have already heard some of these diseases mentioned by Dr. Stack during his talk, but these are the pathogens that cause these diseases. And as you can see, upon introduction of vaccination in the last century specifically, you can see a dramatic decline in the notification of diseases. And that is because vaccines are very, very effective in mitigating spread of disease as well as severity of disease. So I wanted to talk about how in infectious disease progression works. So we have essentially at the beginning exposure to pathogen, and pathogens can be viruses or bacteria, fungi, or parasites. And then following that, you have disease manifestations, some type of symptomatic disease. Symptoms of the disease begin to happen. For example, in the case of the COVID-19, it could be fatigue, it could be difficulty breathing, and headaches, fevers, etc. And at this point, that's usually when people try to go see a doctor, report their symptoms, and there's some kind of medical intervention or treatment course that can happen. So in the case of COVID-19, for instance, we had remdesivir that was being used as a treatment. And following this course, you have some kind of outcome. So these can be really positive, where there is full recovery, or they could be recovery with side effects. We had in the previous panel, Dr. Taylor talking about these sort of long-term side effects that COVID-19 patients have been experiencing. And unfortunately, there can also be death. Now, we have different points of intervention that we can do. I've talked already about medical intervention. Vaccine intervention is really so wonderful because it tackles the disease progression at sort of the initial stages. So it's helping to reduce spread, and it's actually preventing the disease from progressing all the way to severe symptomatic disease. And so it's a much better option, particularly in conditions where treatments are unavailable for diseases. Next slide, please. And so next slide. Thank you. Yes. Okay. And we talk about the benefits of vaccines. So they reduce transmission rates in communities. They reduce severity and long-term negative outcomes, as I've already touched on in the previous slide. They reduce case fatality rates. And what we mean is you actually have reduced mortality rates because of vaccines. Furthermore, there is a prevention of emergence of more virulent and pathogenic strains. And I think the emergence of the Delta variant of the SARS-CoV-2 virus is a key lesson for us in this. And finally, vaccines are proven in a safe way to generate an effective immune response. Now, I just want to go through some epidemiological terms here and talk to you about disease progression on a population level, so you can really get a better sense of the power of vaccines. So normally, when we know the rate at which disease spreads in a community for a specific disease, those are known to be sort of endemic because they spread at a known rate. And because we know the disease well, we can contain it to some extent. Sometimes you have sporadic cases that emerge in various areas as well. And then finally, you have progression into an epidemic or an outbreak if the cases begin to rise significantly above the known and predicted rates. And then finally, when this begins to extend over larger geographical areas and becomes a global condition, then it's known as a pandemic. And the power of vaccines is that they're sort of like a threshold that helps us to keep the spread of that disease at a threshold where we can predict and control what's going on. And you know, as most of you have been following the news, that the Delta virus has essentially overwhelmed the healthcare system, and that's because we have been unable to contain it. And vaccines are pretty much the best option that we have right now to address some of these problems. Next slide, please. So here is where I want to venture out into talking about vaccine research, development, approval process. And so I've divided this part of the talk into two sections. So we're going to have the first bit, which is the research and discovery, as well as testing in pre-clinical trials, and that involves using animal models. And then finally, there's the testing that goes on in human subjects, and that's the clinical trial stage. And I want to emphasize that in all of these stages, the vaccines must be shown to be safe. They must be shown to have immunogenicity. And what we mean by that is that we need to actually show that the vaccine is inducing an immune response. And I'll talk about how we do that in a few slides here. And finally, protective efficacy. And that means that we need to show the vaccines are actually helping to reduce severity of disease in individuals. And finally, in the clinical trial stage, we actually progress from very healthy adults to healthy adolescents or teenagers, and then we progress out into more vulnerable populations. So if the vaccine has been shown to be unsafe in preclinical trials, there's no progression into the clinical trial phase. And if the initial clinical trials show that the vaccine is unsafe even for healthy individuals, then there's absolutely no progression moving forward. So I want to highlight that important part of the clinical trial stage. Next slide, please. So in the research and discovery state, this is where we're developing the vaccine candidate. So this could be an improved version of an existing vaccine. It could be a vaccine with a new mechanism of action. And so once that novel vaccine has been developed, then we test it in clinical trials. And we're going to walk through each of these levels one by one. Next slide. So we have the development stage, and these vaccines can be live or inactivated. And what we mean by that is the live vaccines are attenuated or weakened forms of the disease-causing pathogen, whereas the inactivated vaccines are only using parts of the pathogen or some aspect of the pathogen in order to function as a vaccine. Now, the difference between the two is that in live vaccines, you always have some small chance, very, very small chance of uncontrolled replication occurring, particularly in immunocompromised individuals, whereas that risk is basically absent in inactive viruses. And I wanted to emphasize here that some of the vaccines which most of us have taken, for example, for measles or polio, rubella, all of these are sort of either killed whole organism or attenuated live pathogens that are used to elicit an immune response. However, we have these other categories of vaccines that have emerged in recent years, and these particular vaccines only use some aspect of the pathogen. They are inactive. And here I've highlighted, because this is of great interest to us in light of the COVID-19 pandemic, the nucleic acid vaccine. And so it has a lipid coat and it has an mRNA in there. And that mRNA actually codes for only part of the pathogen, not the entire virus, just the part that is known as a spike protein. And so that's what helps the immune system to ramp up and prepare for potential infection with SARS-CoV-2. And so I want to highlight that, in fact, this mRNA-based vaccine is in the inactivated vaccines category. It's actually one of the safest vaccines we've generated in the past few years. Next slide, please. So following the generation or the production of a vaccine candidate, we then go to the stage where we test in preclinical trials. And in this stage, we're really trying to connect the vaccine with immune response. And finally, we're trying to check for any adverse effects. And right here, I want to pause and point out that I will be trying to reference a lot of the articles that I used in order to present the data to you. So at the bottom right, at the bottom left or the bottom right, you should be able to see these references, and you can use those references later on for yourself to look at some of this data. Now, in this preclinical trial stage for the Pfizer and BioNTech vaccine, for instance, these vaccines were tested in rhesus macaques as well as mice before progressing into any clinical trial stage. And when they were testing in animal models, what they found was not only is the vaccine safe, but in fact, they noticed that the macaques or the primates that had been vaccinated had a much lower viral load. And so the vaccine was safe and it looked like it was working really well. So that was excellent data that allowed them to then move on to the next phase, which is the clinical trial stage. Next slide, please. So in the clinical trial stage, we're looking at testing the vaccine in various human volunteers before approval by FDA, and then also monitoring of the vaccine even after it has been approved. And I want to point out that at times you may see not just sort of a clear-cut phase 1 or clear-cut phase 2, but because some of these trials fulfill the requirements of both phase 1 or phase 2 trials, you might see them referred to as phase 1, 2, or phase 2, 3. And once again, I want to emphasize that we're looking for safety, we're looking for immune response, and we're looking for protective efficacy. Next slide, please. So in all clinical trials, and unfortunately, this is where the animations come in, so I'll try to simply talk about the requirements for each step of the clinical trials. Number one, they must be safe and they must be non-toxic. So people who are involved in the clinical trials are actually, oftentimes people try to have the vaccinations going on near hospital sites so they can monitor how their volunteers are responding to vaccinations, and they also must be non-toxic. And here is a resource for you from, this is straight from the FDA website. There's actually a guidance on non-toxicity. And so these are graded 1 through 4, ranging from mild to potentially life-threatening. And any new vaccination vaccinations that are being tested must abide by these rules. The other bit is that the testing must be thorough. It must limit bias. So for instance, if the people developing the vaccine and testing it decide to make any changes to the formulation of the vaccine, these have to be tested once again through clinical trials. They can't simply make random changes in the middle of clinical trial progress. And finally, I'd like to point out that we very much try to limit bias in those clinical trial stages. And what that means is we do observer blinded experiments where people who are participating in the clinical trials, as well as people who are administering the doses, and really a lot of people who are involved in looking at the data, are blinded to who is receiving the vaccine and who is receiving placebo, for instance. And so that's really helping us to limit bias. Next slide, please. And I wanted to give you some resources here. I'd like to let you know that a lot of the articles that have been published on vaccine development are actually open access. And this is deliberate because this is something that has affected all of our lives. So you can actually find this data published by the scientists online if you just do keyword searches, and you can read them for yourself. And as I said, the data I'm referencing, I will have on my slides, so you can always go back and look those up for yourself. And finally, you can actually go to clinicaltrials.gov and you can use keyword searches, or you can use the clinical trial number if you have that information. And if you enter that, you have access to a lot of information about the clinical trial, the number of participants involved, the locations of the trials, etc. So I just want to let you know that this is available for you to access. So feel free to look at that in your spare time if you feel so inclined. Next slide, please. So the first phase is where we're testing safety in humans. And the key thing is to establish that the vaccine is safe. And as I mentioned, it must have already been tested in preclinical trials. And because the primary goal is to establish safety, we're testing it first in healthy, immunocompetent volunteers, people who don't have any underlying diseases, who have not had any illnesses before, and of course, who have not been exposed to the pathogen that we are trying to develop a vaccine against. And so this is where the administrators or the ones who are developing the vaccine are really beginning to sort of test it on the ground in individual volunteers. And I just want to emphasize here that unlike drugs that are used to treat diseased individuals or people who are sick, vaccines are actually administered to healthy individuals to prevent getting sick. And so the safety bar that the FDA has set for this is actually very high because of that reason. Next slide, please. And so here's an example. What I will do as I talk about these trials is I will talk about the objectives that we have for each phase, and then I'll give you an example using the Pfizer BioNTech mRNA vaccine. So you can look at these side by side and really get a better sense of how well these vaccines were actually hitting the benchmark for each of the requirements within each phase. So in the first phase, as I mentioned, the key thing is to establish safety. So we're looking at what kind of adverse reactions are there. We're collecting data on immune response, specifically the adaptive immune response, where you have antibodies, neutralizing antibodies being generated, as well as T-cell response. And we're also trying to test the conditions for how to administer the vaccine. The test group for this is usually small because it's the initial trial and we're trying to see how safe it is. And the age group is usually members of the target population. So we're trying to select individuals who are part of the population that would need to get vaccinated once the vaccine is approved. So you can see here, for example, in the case of safety, that from the beginning, the people administering the dose and monitoring for safety noticed that there was injection at pain site, there was fatigue, also headaches, and even some fever associated with giving the vaccine. But this wore out within about a week or so of receiving the vaccine. Not only that, this is actually proof that the immune system is working. It's responding to a foreign agent in the body. So they were taking careful record and keeping track of these phenomena. Finally, they were looking at immune response, looking at any neutralizing antibodies that were observed, and they did notice that antibodies were being made in order to fight off the disease. Finally, they tested different amounts of the vaccine. They tested giving doses at different day intervals, and they also had a pretty high number of participants, 195, and they gave some placebo, gave some the vaccine to notice if there were, to record whether there were any differences in safety. And they also tested the vaccine in older individuals as well as younger ones. And there's reason for this. You'll recall that in the early days, especially with the alpha variant, the spread could happen even with young individuals, though the severity of the disease was not as strong. They could still spread the virus. And so really, in order to achieve herd immunity, a large number of the population would need to be vaccinated, which is why they were testing these different individuals. Next slide, please. And in phase 2, we're kind of still testing the same things that we were in phase 1. So you'll notice that there's actually a good deal of overlap between phase 1 and 2 trials. So testing for safety, immunogenicity, you usually have a higher number of participants. You're still collecting tons of information on the kinds of reactions you're getting from the volunteers when you're dosing them with the vaccines. And we're also looking at blinded experiments. So we're trying to make sure that anyone involved in the administration of the vaccine, be it participants or the people who are administering the vaccine, are blinded to who's getting the vaccine, who's getting a placebo, or who's getting a control. That way, there is as little bias in analyzing the data as possible. Next slide, please. Okay, so here's an example of what that looks like in phase 2. So we're determining optimal dose. And if the data looks really good from phase 1 and 2, we can actually begin to plan for phase 3 trials. You have usually a higher number of participants, but it could also kind of be low, 50 to about 500, depending on the test itself. And then the age group is similar to what we would have tested in phase 1 trials. So in this case, you can see that they're testing a fewer number of doses because they've kind of looked at different alternatives already in the first stage, in the first phase. And they also tried to see, well, instead of giving 2 doses, could we get away with eliciting the needed amount of immune response by just giving a higher amount of 1 dose? And they found that actually that didn't work as well as giving 2 doses in 21-day intervals, which is why to this day we are taking 2 doses of the vaccine. And so they had about 76 participants, and the age group they tested here was about 19 to 54. And so this is sort of like a phase 1-2 because it's meeting the requirements of both kinds of clinical trials. Next slide, please. And I want to let you know that in the phase 1-2 clinical trials, which is the initial safety trials in target populations, they were able to see excellent T-cell response. So that means the immune system was being activated. They also saw that the vaccine was tolerated very well. In fact, there was some participants there who had a fever. And once again, this wore away within days. So the reaction was actually within the requirements of the FDA for non-toxicity and safety of vaccines. And finally, this is when they began to register people for testing in the phase 2-3 trials, as well, as I mentioned in the previous slide, of course, given that the phase 1 and 2 trials went well and were validated. Next slide, please. In the phase 3, or the protective efficacy trial, this is where we're really looking at how good is the virus at reducing disease severity. And so since we've already established safety as well as immune response in earlier clinical trials, in this stage, we're really trying to see, okay, is this good? How good is it at reducing disease severity? And you can also get some of that information to a good degree in the phase 1 and 2 trials, but this is where you get a much better sense of protective efficacy because you're testing in larger numbers of participants. So you can have about 1,000 to about 150,000 participants here. And finally, these are large trials that can go on for years. And if you have sufficient data, you can actually apply after phase 3 trials for approval of use of the vaccine with the FDA. Next slide, please. And I want to talk here about the difference between vaccine efficacy and effectiveness. It's really important that we understand what this means. So vaccine efficacy is actually, it refers to how the vaccine performs in ideal conditions. So it's within the testing conditions of the phase 3. And effectiveness, vaccine effectiveness is how well this translates into the wider population once the vaccine begins to administered. So when we say, for example, that a vaccine is 80%, has 80% efficacy, we are not saying that the vaccine will only work 80% of the time. It actually means that 80% fewer people will contract the disease. Or in other words, there's 80% reduced risk in disease severity for individuals who might have had really severe disease if they had not been vaccinated. Next slide, please. And so here's the example for phase 3 trials. In the case of the Pfizer Moderna vaccine, BioNTech vaccine, they had about over 43,000 participants. Half received vaccine, half received a placebo, and they had about 16-year-olds as well as older groups involved in the vaccine. And this is where the vaccine was tested in multi-site locations. So it was sort of like a global endeavor that happened. Some of the people that were involved in the test had to be eliminated because they found out that they were either exposed to the virus or had the disease. And so that kind of messed with the data. So they had to eliminate some numbers there. That's absolutely normal. But still, they had over 30,000 in each category that they could use for their data. Next slide, please. And what they found was incredible because they saw that there was 95% efficacy. So the capacity of this vaccine to reduce disease severity was incredible. And the safety profile did not change when they tested this in larger numbers of population. It was still pain at the injection site, fatigue, and headache. And once again, these were all symptoms that faded away within about 7 to 9 days or so of receiving the vaccine. Next slide. And so after that, they were able to submit a BLA to the FDA, at which point it got approved. And here's the information that you would need. Once again, I apologize. I know you can't read what's behind these because those would have been animated. But the essential requirements for BLA is manufacturing information. So the FDA needs to see how this vaccine is getting manufactured. They also need the data from the preclinical and clinical trial information that they can use in order to approve. And you'll know that last month, this vaccine was finally approved by the FDA. Now, I want to underscore that approval by the FDA does not necessarily mean that there aren't sub-studies or ongoing studies that are associated with the vaccine. So the approval is very conditional. So if the data is sufficient for administering the vaccine to a specific group of people, it would be approved by the FDA for that. But that doesn't mean that there aren't ongoing studies for other population groups. And those population groups cannot receive the vaccine unless there is some clear indication by the HHS that they can do so. And I'll talk about exceptions to that here in a little bit. Go ahead, please. And phase 4, this is the part where we're looking at vaccine effectiveness. The vaccine is already being administered to the population. We're collecting data on how this is affecting populations at a wider scale. And as I mentioned before, trials can be ongoing. For instance, with the Pfizer BioNTech mRNA vaccine, there are trials that are still going on for ages 12 and under. And some new developments too for children for ages 12 to 15-year-old. Next slide, please. And I want to talk about the ACIP here, as well as the FDA, and how they work together very strongly to help ensure that the vaccines are safe and that risk is mitigated. So I want to assure you that there's these two independent bodies here. They work together, they collaborate, but they vet the vaccine rigorously to ensure that there is no danger. And there are, in all of these, specifically the ACIP members who are experts in their areas to ensure that that happens. Next slide, please. Next slide. So we're going to talk about when emergencies occur. And once again, this is where the EUA comes in. So emergency use authorization can be declared by the Secretary of HHS in the case of a public emergency or a potential public emergency. And that's what happened on the 4th of February last year. Next slide. And you can see that once the vaccine, the clinical phase 3 study was published on the 18th of November, the FDA issued an EUA for the COVID-19 vaccine because the phase 3 clinical trial data was so good. And I want to remind all of us that on that day, the cases were still coming in pretty strong. The US had a third of the new cases as well as 3,000 deaths on that day. So the benefits associated with simply approving the vaccine were extremely high. Next slide. And here are some of the factors that can affect vaccine, the speed of vaccine approval, whether or not you have adequate numbers of people to meet the requirements of phase 1 to 3 clinical trials, whether you have single or multi-center trials going on. So if they're multi-center, they might take maybe a little bit longer time. Cost. That's the CARES Act really helped to fund some of the vaccine research with some of these companies and helped them to fast track vaccine production. There's collaboration, massive collaboration across the globe. For instance, phase 1 clinical trials had already been concluded in Germany before they started in the US. So that data was really useful for clinical trial studies here. And finally, EUAs were really, really useful. So even before the vaccine got approved by the FDA, the ACIP as well as the FDA looked at the clinical trial data and agreed that there was so much benefit to the vaccine that they should be administered to individuals ages 16 and older. And currently, it is being used under EUA for 12 to 15-year-olds. And that's based on efficacy data on tests that were done with about 2,300 children. And currently, this vaccine is still being studied for ages 12 and under. And I just wanted to give you an example using that about why there was such a fast tracking of this vaccine in the last year. Next slide. And in conclusion, vaccines are biological products that confer protection against symptomatic disease, infectious diseases, and specifically speaking. And before authorization by the FDA, vaccines undergo rigorous safety, immunogenicity, and protective efficacy tests through the phase 1 to 3 clinical trials. Vaccines, even after being approved for use, are monitored closely by the FDA and the ACIP, as well as the vaccine manufacturers, even after licensure and approval for use. That's really important. And finally, we have EUAs that can be declared during public health emergencies or potential public health emergencies in order to speed the rate at which vaccines can be authorized for use by the FDA. And so I'll stop right there. Thank you so much for your time and for working with us with our technical difficulties, and I'll hand it over to you guys.
That was wonderful. You crammed in a great deal of information, Dr. Hankore. We appreciate it so very much. And